Okay, so welcome back to this next video in which we are discussing the SREBP pathway. Okay, right, so we're just currently in the process of discussing how cells can acquire cholesterol, and really I think it's very important to firstly go over this before we see the SREBP pathway, because the SREBP pathway is going to affect these ways of taking up cholesterol. Okay, so if we don't know the ways of taking up cholesterol, then it's pointless looking at the SREBP pathway. Okay, right. We have discussed that the LDL receptor has bound to the LDL here, and that has triggered some sort of conformational change in the LDL receptor, which leads to the exposure of this endocytic MPXY motif. Okay, and this MPXY motif will then be recognized by a cytoplasmic protein known as the LDL receptor adapter protein 1, okay, the LDL-RAP1, or also called the autosomal recessive hypercholesterolemia protein, the ARH protein. Now, the ARH is truly an adapter protein because it will bind to the receptor, and then it's also going to bind to clathrin, and then clathrin is going to be responsible for endocytosing this vesicle. So let me now discuss clathrin with you. So I'll get a new piece of paper, and then we'll discuss clathrin, then we'll get to the endocytosis. So basically, clathrin is made up of two proteins, okay? There is a clathrin heavy chain and there is a clathrin light chain. So let me show you the clathrin heavy chain firstly. So the clathrin heavy chain is often schematically drawn like so. Okay, so this portion right up here, this is known as the N-terminal domain of the clathrin heavy chain. Okay, so the N-terminal domain. This portion, in fact I might colour it in, so we'll have the N-terminal domain in vivid purple here. This portion in the middle here, this is known as the distal domain. Okay, so this is the distal domain, and you'll see why these names are appropriate in a moment when we look at the structure of a clathrin triskelion. Okay, so in blue here, this is the distal domain. And then, here, we have the proximal domain of the clathrin heavy chain. So this is the proximal domain, okay? And we'll colour in this proximal domain of the clathrin heavy chain in light green here. So this portion is all the proximal domain. And then this final portion, which I'm going to colour in orange here, this is the trimerization domain. So this is the trimerization domain. Okay, right. So that's the different parts of the clathrin heavy chain then. So this is all one protein, and this is the clathrin heavy chain. So what's going to happen is that clathrin heavy chains will associate with clathrin light chains to make a full clathrin molecule, and then we're going to bind 3 full clathrin molecules together to make a clathrin triskelion. Okay, so to convert a clathrin heavy chain into a clathrin molecule, what we need to do is bind on the clathrin light chain here. So what colour should I colour that in? Turquoise. In turquoise here, this little protein sitting alongside the clathrin heavy chain, this is going to represent the clathrin light chain. So this is the clathrin light chain, and the full thing now, a clathrin heavy chain with a clathrin light chain bound to it, is a clathrin molecule. And what we're going to do is we're going to stick 3 of these together to make the normal form that clathrin exists in within the cell, which is a clathrin triskelion. Okay, so let me show you the structure of a clathrin triskelion then. Okay, right, so what you're going to do is basically take 3 of these clathrin molecules, and you're going to trimerize them together by binding them at the trimerization domains of the heavy chains. So let me show this. So here is our first clathrin heavy chain, like so. It's got a clathrin light chain associated with it, and then you're also going to have another clathrin heavy chain here, which will again have a clathrin light chain associated with it. Then another clathrin heavy chain here, which will again have a clathrin light chain associated with it. Okay, so this is the structure of a clathrin triskelion, and 3 clathrin molecules have been bound together via the tetramization, sorry, not the tetramization, I'm thinking of potassium channels, the trimerization domain of these clathrin heavy chains here. Okay, right, and this is the structure known as a clathrin triskelion. Okay, so what's going to happen is clathrin triskelions will come and bind to the autosomal recessive hypercholesterolemia protein, also known as the LDL receptor adapter protein 1, and they will then start polymerizing. So I want to show you, before I actually draw them attaching to the adapter protein, I want to show you how clathrin triskelions can polymerize. So basically they can polymerize into a buckyball-like structure. Okay, so if you've seen a football, it's going to be like that. Okay, so the same sort of shape as Buckminster fullerene. So I'll have a go at drawing a bit of this. So we'll start off with a regular pentagon which is drawn in the plane. So imagine this regular pentagon is within the plane. Okay, now coming off the regular pentagon, you then have regular hexagons. Okay, now these are not going to stay within the plane. These are going to go into the page away from us. Okay, so imagine that these regular hexagons are now bending into the page away from us. It would be an interesting morning's work just to make one of these out of paper, I suppose. If you were a mathematician, maybe you'd do it. So basically, coming off this pentagon, you're going to have 5 of these regular hexagons going into the page. Okay, like so. So here's another one. They're all going into the page. They are not, these hexagons are not in the plane of the page. They're bent inwards. Okay, like so. And then here's another one here. Okay, so all of that is sort of curved over, so it's sort of like forming the top of a dome, if you like. So the pentagon is in not the same plane as the paper, but the hexagons are all folding back down. Okay, and then what you'll do is in these gaps here, which we've now got 1, 2, 3, 4, 5 of, you're going to now put little regular pentagons. So we'll have a regular pentagon in there, a regular pentagon in there, and these will almost now be vertical, these regular pentagons that are being put in here. Okay, there's another regular pentagon, here's another regular pentagon, there's another regular pentagon there. And then around the regular pentagons here, each of these, you're going to make sure that there are 5 hexagons, and you'll continue this on, and eventually it will form a beautiful sphere, basically. And that's the structure of Buckminster fullerene. So, what's going to happen is these clathrin triskelions are going to, Buckminster fullerene is the chemical made out of carbon that has this sort of shape. The shape, this sort of tessellation of pentagons and hexagons is just called a buckyball. Okay, it's the same shape as a football. If you've seen or a soccer ball if you're American. If you've seen a football, then it's that pattern. Okay, so these clathrin triskelions are going to join together to make one of these buckyballs. Okay, so let me try and show you how this is going to work. So it's not going to work perfectly. You might hope that, is it actually going to work perfectly, that you won't have a single bit of overlap? No. Basically, clathrin triskelions will be overlapping when they're making this structure. So let me just indicate where a clathrin triskelion could be. So for instance, here could be a clathrin heavy chain. Okay, and let's put the clathrin light chain on here in red, or vivid purple rather. So there is a clathrin within here. So let's now turn it into a clathrin triskelion. So off here we'll have another clathrin heavy chain, and off here we'll have another clathrin heavy chain, like so. Okay, so there's a clathrin triskelion, and let me now just add in the light chains, 1, 2, 3. Okay, so that's a clathrin triskelion making up part of this buckyball structure. And now, you might put another one in and it won't be perfectly non-overlapping. So we could put another one centered here where, the clathrin molecule heavy chain would have its trimerization domain here, then its proximal domain here, distal domain here, and then its N-terminal domain here, so that it was overlapping with this other clathrin triskelion. So it doesn't work perfectly, but it does form this polymer that is in this buckyball shape. Okay, right. So, back to the endocytosis of LDL receptors then. So at the moment we've got our plasma membrane here, we've got our LDL receptor here, which has got LDL bound to it, okay, via the apolipoprotein B100 here. So this can represent the apolipoprotein B100, this turquoise blob here. And then we've got the LDL receptor ligand binding domain here in blue. Okay, then we have this special domain on the cytoplasmic side, which was the NPXY motif, and then we have the carboxylic acid terminus here. And now bound to that NPXY motif, what you have is the autosomal recessive hypercholesterolemia protein, the ARH. Okay, and now the ARH can bind to clathrin, so a clathrin molecule will come and bind here, and I'm going to demote clathrin to being represented just as a blob now rather than as a triskelion like so, but you should think of it as a triskelion. So here comes a clathrin molecule in orange, and then what's going to start happening is loads of other clathrin molecules will attach to that, and they'll produce this buckyball structure. And as they produce this buckyball structure, what's going to happen is they will try and produce it, and they're going to pull the membrane down with it, basically. So let me try and show this. So basically what you're going to end up with is something that looks like so. Okay, so here is our endocytic vesicle being formed here. Then we have our LDL receptor with its ligand binding domain here. Here is the LDL molecule bound to the LDL receptor. Okay, and here is the apolipoprotein B100 in turquoise here. Then in blue we have the LDL receptor ligand binding domain. Then here we have the NPXY motif, so this is the NPXY motif, and then we have the carboxylic acid terminus here. Then attached to the NPXY motif we have the autosomal recessive hypercholesterolemia protein, and then polymerizing on from this you then have this clathrin buckyball forming, and it's attached to the autosomal recessive hypercholesterolemia protein. So I'll try and draw this like so. So starting to get this polymer forming here, and I'm not going to actually draw it as a buckyball, I'm not going to attempt that. But, oh, it's smudged, that's not nice, but never mind. Okay, so you've got clathrin molecule after clathrin molecule after clathrin molecule after clathrin molecule. All of them are polymerizing together to make this sort of buckyball structure that is going to be pulling the membrane inwards like so. Okay, so all of this is clathrin. Okay, so I'll colour it all in orange. Okay, right, and what's going to eventually happen is that you're going to complete this buckyball, so the clathrin will continue to polymerize, and it will complete the buckyball, and then what you'll end up with is a completely pinched off vesicle, basically, which is coated in clathrin. So let me draw this next. So here is the clathrin continuing on, like so. So what you'll end up with then, overall, is a vesicle that's been pinched off from the plasma membrane. So here is the vesicle, and then it's now surrounded by this clathrin coat, which I'll show here. So this is representing the clathrin coat, and the clathrin coat is still attached to the LDL receptor via this adapter protein. So we've got many, many clathrin proteins all having been polymerized together to make this complete buckyball that surrounds the vesicle. Okay, right, there we go. And then we have the LDL receptor here with its ligand binding domain here, and then the LDL is still bound here via the apolipoprotein B100, and then we have our adapter protein there, which is the LDL receptor adapter protein 1. So I'll colour in the LDL receptor adapter protein 1 in purple. So this is the autosomal recessive hypercholesterolemia protein. And then the clathrin coat can be in orange here. So this is the clathrin coat. Okay, right. And then I'll colour also in the ligand binding domain of the LDL receptor in blue and the apolipoprotein B100 in turquoise. Okay, and this pen's starting to go, I think. Okay, so there's our clathrin coat in orange. Then we've got the ligand binding domain of the LDL receptor in blue and the apolipoprotein B100 in turquoise. So that's how we endocytose the LDL receptor with the LDL cargo attached to it. So this at the moment is a clathrin coated endocytic vesicle. Okay, so it's a vesicle that we have just got via an endocytic process. So a clathrin-coated endocytic vesicle. Now, you can't do anything with this clathrin-coated endocytic vesicle whilst it is still coated in clathrin. So what you need to do is remove the clathrin coat. So the clathrin molecules will be removed, the adapter protein will also be removed, and what you'll then end up with is a vesicle, like so, which has the LDL receptor within it. So here is our LDL receptor, here's the ligand binding domain, and then it's bound to its LDL cargo here via the apolipoprotein B100. So here is the LDL receptor ligand binding domain in blue, and here is the apolipoprotein B100 in turquoise. Okay, so we now have our endocytic vesicle. Now what's going to happen is this endocytic vesicle is going to go to an intracellular organelle called the early endosome. Okay, so let's now discuss the endocytic pathway, where this LDL receptor with cargo containing endocytic vesicle is going to go. Okay, so let's draw our endocytic vesicle here, which has now lost its clathrin coat. Okay, so here is the LDL receptor with its cargo bound to it, and basically it's going to now go to the early endosome. Now the early endosome is a large intracellular organelle compared to the endocytic vesicle. Okay, so it's a much bigger structure. So here, let's say this structure here is the early endosome. Okay, and what's going to happen is this little endocytic vesicle here is going to come along and fuse with the membrane of the early endosome. Okay, so in comes the endocytic vesicle, and it's going to fuse with the membrane of the early endosome. Okay, and what then is going to happen is that the LDL receptor will have its ligand binding domain facing into the lumen of the early endosome, and bound to the ligand binding domain of the LDL receptor you'll have the apolipoprotein B100. Okay, so here is the apolipoprotein B100, and here is the ligand binding domain of the LDL receptor. So now our LDL receptor and cargo are within the early endosome. Now, it doesn't remain in the early endosome for long. What will happen is you'll pinch off another vesicle off the early endosome, which will contain this LDL receptor along with its cargo once again, and this vesicle is going to go to another intracellular organelle, which is called the late endosome. Okay, so here's the ligand binding domain of the LDL receptor. Here's the apolipoprotein B100. Now, this little vesicle is going to transport the LDL receptor along with its cargo from the early endosome to the late endosome here. Okay, so this little vesicle is going to come along and fuse with the membrane of the late endosome, and then here is the LDL receptor now within the late endosome, and it's got its LDL cargo, at least initially, bound to it. Okay, now something more interesting is going to happen within the late endosome than happened in the early endosome. Okay, the late endosome has an extremely acidic pH, so a very high concentration of protons. Now, if you remember, how did the LDL receptor bind to the apolipoprotein B100? Well, the LDL receptor ligand binding domain had a number of acidic residues which donated their protons away and therefore got a negative charge. Meanwhile, the apolipoprotein B100 had a number of basic residues which gained protons and therefore gained a positive charge, and the negative charge bound to the positive charge, and that was what was holding the apolipoprotein B100 to the ligand binding domain of the LDL receptor. Now, if we've got a very high proton concentration, okay, so we've got a very low pH, remember, pH goes down as proton concentration goes up, okay, because it's a negative log scale. Okay, basically what's going to happen to those acidic residues on the ligand binding domain of the LDL receptor is that they're all going to get protonated. Okay, so remember, the acidic residues originally gave away their protons and got a negative charge when they became their conjugate bases. If there's a very high proton concentration, all of those acidic residues are going to have their protons attached to them, i.e. they won't give them away to the solution, or rather, they might give them away, but the instant they do, they'll get another proton attached to them, which means they will be neutral, okay? So they won't be now able to interact with the positive charge of the basic residues of the apolipoprotein B100. So the overall result of this low pH within the late endosome is that the LDL and the LDL receptor are going to part, okay? So they're going to fall apart. So here is the apolipoprotein B100, and it's now no longer bound to the ligand binding domain of the LDL receptor. Now, what's then going to happen is that a vesicle will bud off the late endosome, okay? And this will take the LDL receptor, which now no longer has the LDL bound to it, back to the plasma membrane. So this is going to recycle the LDL receptor, basically. So this will go back to the plasma membrane here, and you'll end up putting the LDL receptor back on the plasma membrane. So that's recycling of the LDL receptor back to the plasma membrane. Okay, meanwhile, another vesicle will bud off the late endosome, which will contain the LDL particle. Okay, so here is our LDL particle with this apolipoprotein B100 within its phospholipid monomer here. So this is apolipoprotein B100. Okay, and what's then going to happen is this little vesicle is going to go and fuse with another vesicle called a lysosome. Okay, and lysosomes contain a huge number of enzymes which will break down the components of the LDL molecule. Okay, so this is going to go and fuse with this lysosome here. And basically what's going to happen is the components of the LDL molecule are going to get broken down. So for instance, the apolipoprotein B100 will be broken down into amino acids, okay, or at least peptide fragments, small fragments. So let's just remind ourselves of the structure of the LDL molecule in a little bit more detail. So remember, we had the phospholipid monomer here, which mainly consisted of lecithin molecules. Okay, we then had our apolipoprotein B100. We've discussed what's going to happen to the apolipoprotein B100. So let's now go on to the lecithin molecules. So the phosphatidylcholine, which makes up most of the phospholipid monomer here, these are going to be broken down within the lysosome. Okay, so you'll release free fatty acids and glycerol and things like that. Okay, the cholesterol molecules that are within the phospholipid monomer and are free, those will not be broken down. Those will be allowed to leave the lysosome and go into the cytoplasm. So cholesterol is spared. Okay. Meanwhile, the cholesterol esters, which are, remember, the main cargo which we have within the lipid core of the LDL molecule here, these will be broken down. You will break that ester link that attaches the long-chain carboxylic acid to the cholesterol molecule. So you'll break that, and you'll get a free fatty acid molecule being produced, and yet more cholesterol. So the main message here is that we are delivering cholesterol to the cytoplasm of the cell. So that now concludes how cells can get cholesterol via absorption of cholesterol through absorption of these LDL molecules. Okay, now what we want to turn our attention to is how they can also produce cholesterol via synthesis. Okay, now we're not going to go through the entire biosynthetic pathway of cholesterol because it takes ages. We're just going to look at one important reaction, the rate-limiting step in cholesterol biosynthesis. So basically, in cholesterol biosynthesis, you start with acetyl coenzyme A, and you then eventually end up with a cholesterol molecule, and the entire process occurs within the cytoplasm of the cell. Now, I am going to tell you about a single reaction within the cholesterol biosynthesis pathway, and if this reaction is blocked, you cannot synthesize cholesterol. Okay, and this is actually the site of action of the incredibly successful drugs, statins. They block the enzyme that catalyzes this reaction and therefore block cholesterol biosynthesis within cells. So the only source of cholesterol the body can then have is dietary cholesterol. Okay, right. Let me now show you the reaction that is so important. So basically, it's going to involve a molecule called HMG-CoA. Okay, and HMG-CoA is short for beta-hydroxy, beta-methyl, so the H is for hydroxy here, then we have beta-methyl, the M is for methyl, and then the G is for glutaryl, and then the CoA is for, glutaryl, then the CoA is for coenzyme A. Okay, so this is beta-hydroxy, beta-methyl, glutaryl, coenzyme A. So, basically, we need to look at the different parts of this name in turn. We'll start off with what glutaric acid is. So basically, glutaric acid is a 5-carbon carboxylic acid molecule. 1, 2, 3, 4, 5. But it's better than that. It's actually got 2 carboxylic acid groups, one at each end. So this is going to be a carboxylic acid group. This is also going to be a carboxylic acid group. Okay, so I will draw half of them. Okay, so I'll draw their carbonyl groups, because basically one of these carboxylic acid groups is going to be involved in a link known as a thioester link to coenzyme A. In fact, I might draw the alcohol group here and then I'll show doing the thioester link. So, we are going to link this carboxylic acid group here to coenzyme A. Now, all we need to know about the structure of coenzyme A is that it has a thiol group. So I'm just going to draw a box and put CoA in there, and then I'm going to put this thiol group off the side. Okay, now thiol groups are actually very similar to alcohol groups. Sulfur is in the same group of the periodic table as oxygen. If we replace that sulfur atom with an oxygen, this would be an alcohol group. Sulfur, being in the same group of the periodic table, has very similar chemical properties to oxygen. Therefore, we would expect this thiol group, as it's called, to have very similar properties to an alcohol group. Okay, and indeed it does. And what it can do is it can react with carboxylic acid groups in much the same way as an alcohol group can. So, what you're going to do is, we're familiar with this now, you can take the alcohol group off the carboxylic acid molecule, you can take the hydrogen off the thiol group, you can combine those together to make water, and then you can bind this carbon here of the carboxylic acid group to this sulfur atom of the coenzyme A molecule, and that's sort of a link there where you've got the carbon atom of a carboxylic acid molecule linked to the sulfur atom of a thiol group. That's not called an ester link, but instead it's called a thioester link. Okay, so to draw the parallels between how similar it is to esterification. Okay, so one of these carboxylic acid groups will be involved in this thioester link to coenzyme A. That's the glutaryl coenzyme A. The other one, generally under physiological pH, will have lost its proton, so it will be present just as an oxygen with a negative charge rather than the entire alcohol group here. Now, that's the glutaryl coenzyme A handled. We then need to get some sort of handling of beta-hydroxy, beta-methyl. So basically, the first carbon off the carboxylic acid group of the glutaryl molecule is called the alpha carbon. So this will be called the alpha carbon. The second one will then be called the beta carbon. Okay, so off this beta carbon, we're going to have a hydroxy group, and we're also going to have a methyl group. So here's a methyl group, and here's a hydroxy group, like so. And then that's it. That's all that's special about this molecule. We can then saturate up the other carbons with hydrogens. Okay, so this is the structure of beta-hydroxy, beta-methyl, glutaryl coenzyme A, often just called HMG-CoA for short. Okay, so what's going to happen now is we're going to take these HMG-CoA molecules, which are formed during the first stage of cholesterol biosynthesis. So from acetyl coenzyme A, what you do is you attach 3 acetyl coenzyme A molecules together to make this HMG-CoA molecule, and then you're going to use the HMG-CoA molecules to make something else, which will then be used to make cholesterol. Okay, so we're going to see how this is going to be converted into a molecule known as mevalonate. Okay, so, can't go there. Right. So, let me firstly just redraw out my structure of HMG-CoA so that we've got it on this page. So here is our first carboxylic acid group, which is involved in this thioester link to coenzyme A. Here is the second carbon, which is just in a methylene group. The beta carbon, which is has got a methyl group and an alcohol group coming off. then the fourth carbon here, which we could call the gamma carbon if you were so inclined, and then the final carbon, which is involved in this final carboxylic acid group, which usually has lost its proton and therefore will have a negative charge there. So this is the HMG-CoA molecule. Okay, so what's now going to happen is this can be converted into a molecule called mevalonate. Okay, so we're going to convert it into mevalonate. And this conversion is going to be catalyzed by an enzyme called HMG-CoA reductase. Okay, and from the name you can guess what it's going to do. It's going to reduce HMG-CoA. So, what you're going to need for this reaction is you're going to need 2 molecules of NADPH. Okay, so reduced nicotinamide adenine dinucleotide phosphate. So I suppose I should just give you a brief discussion of what nicotinamide adenine dinucleotide phosphate is. So NADP stands for nicotinamide, that's the N. The A then stands for adenine, the D dinucleotide, and then the P phosphate. So let me draw you a little cartoon of what nicotinamide adenine dinucleotide phosphate actually looks like. So basically it's a dinucleotide, it's 2 nucleotides joined together. So what generally do you think of when you think of a nucleotide? Well, you think of a ribose sugar. Okay, so this pentameric ribose sugar here with the 5th carbon coming off up here, with an organic base attached. Okay, we might as well draw adenine because that way we can get part of this structure, and then it would have a phosphate group attached to it. That's what you generally think of when you think of a nucleotide. And indeed you are going to have this within your structure. This is an adenine nucleotide. Okay, and it's basically going to be linked to another nucleotide. So let me put in another nucleotide here. So I'm going to sort of squash it in here. So here is another ribose sugar here with its 5th carbon and a phosphate group coming off, and the structure that you're then going to have attached to the ribose sugar is not an organic base this time, but it is instead nicotinamide. Okay, so I'll put an N there. That's not an organic base, but it can be attached to the ribose sugar. And you're then going to attach these 2 phosphate groups together. So remember, these phosphate groups will be attached to the alcohol groups that come off the 5th carbons of the ribose sugars via phosphoester links, which we've seen already in this video. Each of these phosphate groups will then have another free alcohol group. You're going to react these 2 together to make a phosphoester link between the 2 phosphate groups. Okay, and that's how you've linked these 2 nucleotides together to create a dinucleotide. Okay, so what I have now drawn for you would be called nicotinamide adenine dinucleotide, and that's the molecule that we all know and love as NAD. Okay, however, you can also add a phosphate group onto this structure to create nicotinamide adenine dinucleotide phosphate. So, we're going to add a phosphate group onto the alcohol group that comes off the 2nd carbon of the ribose sugar. So remember, these are ribose sugars, not deoxyribose sugars. So they have an alcohol group that comes off that 2nd carbon, whereas deoxyribose wouldn't. Okay, so we have attached another phosphate group onto that ribose sugar via a phosphoester link. Now, that creates us nicotinamide adenine dinucleotide phosphate, which is usually just abbreviated to NADP. Now, the nicotinamide group that you have attached onto the ribose actually has a positive charge, so the entire molecule therefore has a positive charge. So people often write NADP and then put a positive there to show the positive charge. Now, why are these molecules so important? Well, basically they can accept something called a hydride anion. Okay, so basically a hydrogen atom that has accepted an additional electron. So hydrogen atoms always have a proton in their nucleus, and the main isotope of hydrogen that is found on this planet has no neutrons within its nucleus. You can get hydrogens where they have neutrons. For instance, deuterium has 1 neutron, tritium has 2 neutrons, but the main form is protium, which has only 1 nucleon, and that's a proton and no neutrons. Okay, right. But then they all then have a single electron surrounding them. So that's a hydrogen atom. So here's the proton at the center in red, and here's the electron orbiting around the proton. Okay, now, usually we create hydrogen cations where we strip the proton of its electron, and we therefore end up with a hydrogen cation here, or just called a proton. But you could give this hydrogen atom an additional electron, and when you do that, you create an anion, a negatively charged ion, with which is called the hydride anion here. Okay, so that's what a hydride anion is, and basically these NADP molecules can accept a hydride anion, and when they accept this hydride anion, which is one proton and two electrons, they clearly become neutral because we're mixing a negative charge with a positive charge, and this is called NADPH, or reduced NADP. Okay, so really, these two molecules of NADPH that we're bringing in here are bringing in two hydride anions. Okay, and they will leave without their hydride anions. So we're bringing in two protons and four electrons. Now we want, oops, what have I done here? NADP. Okay, right, so we want to even it out a bit, so we're also going to bring in two protons from the solution, and that means that we will now be bringing in four protons and four electrons, in other words, four hydrogen atoms. So we're now going to use the HMG-CoA and these four hydrogen atoms to create mevalonate. So what are we going to do? Well, this isn't an electronic mechanism, but it's just to help you understand what's on one side of the equation adds up to what's on the other side. So we're going to break this bond between the carbon atom and the sulfur atom of the coenzyme A molecule, and then we're going to send one electron back to the carbon and one back to the sulfur atom. We're then going to bind a hydrogen atom onto the free electron of this carbon and a hydrogen atom onto the free electron of this sulfur atom. What that will recreate for us is the coenzyme A molecule. Okay, so here's the coenzyme A recreated. We've now got this carbon with a double bond to an oxygen and a hydrogen sticking off it. We're not going to stop there. We're also going to break one of the two bonds within that double bond between the carbon and the oxygen atom. Okay, so we're going to break this bond. We'll send one electron back to the carbon, one back to the oxygen. We're then going to use the other two hydrogen atoms, because remember we brought in four. We've used two in the breaking of this bond. We've still got another two to use. So we'll bind one then to this carbon and one to the oxygen. Okay, and that will create us mevalonate. So let me show you this. So most of the molecule is unchanged. All of these four carbons are unchanged. So here's the carboxylic acid group at the other end. Here is the methylene group. Then in the middle we have this carbon with the methyl group and the alcohol group coming off. Then we have another methylene group here. And then finally, on the end, we have a carbon which has two hydrogens coming off it. It got one from the breaking of this bond here, and then it got another from the breaking of that bond, and then it's got this alcohol group coming off, and that is the structure of mevalonate. And this will then go on to be used in the biosynthesis of cholesterol. Okay, so this enzyme, HMG-CoA reductase, in green here, this is the rate-limiting step. The production of mevalonate limits how much cholesterol the cell can actually produce. Okay, so this is the site where we regulate production of cholesterol biosynthetically within cells. Okay, so if cells have a lot of cholesterol in their cytoplasm, they don't want to be making any more. So they're going to inactivate these enzymes basically, and we want to know how. Okay, in addition, this is the site of action for the exogenous drugs which are known as statins. Okay, so these are cardiovascular protective drugs that have become incredibly successful, and people are now suggesting that everyone over 40 should be on them. Okay, so famous examples of statins are simvastatin, is probably the biggest prescribed one nowadays. Atorvastatin is one of the greatest selling drugs ever, and then rosuvastatin is another good example. So they all end in statin, basically. So whenever you see a drug with statin on the end, you know instantly that it's an inhibitor of HMG-CoA reductase. And the advantage of that is that you can stop the body from synthesizing cholesterol de novo. So the only source of cholesterol is then the dietary cholesterol. And if you've got far too high blood cholesterol levels, then stopping synthesis of cholesterol within cells is a very good idea. Okay, right. We'll call it there for this video, and in the next video we'll begin discussing the SREBP pathway and how this pathway is going to regulate the absorption of cholesterol through LDL receptors and also the production of cholesterol de novo. So basically the point of the SREBP pathway is to say we need to couple how much cholesterol we are absorbing through the LDL receptor and making through de novo biosynthesis to how much cholesterol we actually have in our cytoplasm. If we've got a lot of cholesterol in our cytoplasm, we do not want to be synthesizing more cholesterol and we do not want to be absorbing more cholesterol. Whereas if we've got hardly anything, we don't, we want to be making more cholesterol and we want to be absorbing more cholesterol. So basically, the SREBP pathway is how we regulate the two processes that we've now looked at in detail, so that the cholesterol level in the cell determines how much more cholesterol is going to be coming into the cell. Okay, and we'll continue that discussion in the next video.